All right, this will be a short introduction on PhloemTransport. Um, PhloemTransport is going to be how sugars, hormones, amino acids, and some other compounds are moving within the plant. And we think about this as, and I'm going to talk about it right now, let's just focus on sugars. But the source is going to be where the sugars are actually produced, and the sink is going to be the area that has the demand for those sugars. So in the source, we see a net export of sugars or other compounds. And to provide some context and on maybe like an image in your head, what we're thinking about is a mature leaf that's growing pretty fast. So as a leaf is expanding, it's usually a sink, but once it's matured, but it's still photosynthesizing pretty quickly, it's then exporting those sugars to other actively growing tissues. And so a sink then would be a growing tissue with net demand. And I probably shouldn't put growing tissue, but really, it could be a tissue that's not growing, but that then just has some high demand for energy. So that could also be a sink. We often think about, stems aren't photosynthesizing very much themselves, and so that would be a sink. So the mature leaves would be providing sugars to the stem for stem growth. Root tips or anything below ground, of course, it's not photosynthesizing itself, and so it needs sugars from somewhere else. And then new leaves or new growing tissues typically are a high, like a strong sink, if you will. So like a lot of demand for sugars. All right, let's get this clicking. All right, so the forms that sugars are transported in are either sucrose is a common one, other oligosaccharides, which are fairly large complexes of sugars, bigger than sucrose, I should say, and sugar alcohols. Simple sugars, like simpler than sucrose, so we're talking glucose, fructose, those are smaller molecules. Those are more reactive, and so if they're moving from source to sink, it's possible they would get reduced or have some reaction between or on that pathway, and they wouldn't actually make it to the sink. And so these forms of sugars are more stable, and they're gonna definitely make it from the source all the way to the sink. This is primarily what's in the phloem sap. Hormones that are being signaling, that the plant is signaling, could be in the phloem. Amino acids, which are going to be used to build proteins, and some nutrients might be in there. Yeah, so those are the major components of phloem sap. The rate of transport, so the main point I want to make here is how slow phloem transport is relative to xylem transport. So you can see xylem transport is, 6-7 times as fast as the rate of transport in the phloem, although we don't have great data on the rate of phloem transport. So this is a real approximation, but we do know it's slower than xylem transport. And also, well, we'll talk about this we'll talk about more in a second, but in many tissues in many plants, the xylem and phloem, are co-located together. And so there's some interaction between them as they share water, or water moves back and forth, but we don't know much about the coordination of those two. And it makes sense that they would be coordinated to some degree, but there hasn't been a lot of research on that aspect. Okay, let's talk about where these tissues are located in different plants and different parts of the plant. So if we're thinking of woody tissue, the outer layer of the vascular cambium is where the phloem is going to be. So here's a cross section of a small twig. This division here between the less blue area and the more blue area. So this outer area that's stained really dark blue, this is the phloem. And inside here is the xylem. And there's a layer here that's hard for us to distinguish from anything else, but this is the vascular cambium. And so that's where the cells are actively dividing and growing. And so in woody tissue, it produces the xylem to the inside, and the phloem gets produced to the outside. So I just talked a second ago about how xylem and phloem often are co-located. In this case, in woody tissue, that's not the case. All right, in herbaceous tissue, so we're thinking of both herbaceous plants, but also the leaves and stems of herbaceous plants are going to be, the phloem is found inside the vascular bundles. So I'm sorry, this is a little pixely, but um, I'll show you them anyway. So here's a monocot stem, so like a grass stem, and all of these larger circles that are stained a little bit turquoise here have xylem and phloem both located in there. And yeah, so they're co-located in the stems of monocots, but also in dicots. But what you see here is that these large structures on the outside, where there's some blue and some red, those are the vascular bundles. And you've got xylem here and then phloem typically on the outside. So those are the stems, again, co-located in herbaceous tissue. And then if we think about the leaves a little bit, this is a nice cross-section of a monocot leaf, or a clear cross-section, I should say. And each of these large round structures are the vascular bundles. So you can see we've got a large one here, but also this is another vascular bundle. This bluish stained area is the phloem, and then this yellowish area here, that's where the xylem is. Yeah, and then this is not very clear here, but this is the vascular bundle in a dicot leaf. Same thing, where this reddish area is going to be the xylem, and then this bluish turquoise stuff is the phloem. So again, they're co-located in these vascular bundles in herbaceous tissue. And then lastly, in the roots, we have two sections of the root. So the root is divided into the cortex, which is this outer area, and then the stele, S-T-E-L-E, is this inner area. And so in the roots, it's a little more like the wood, in that xylem and phloem are not, I mean, they're closer together than in the woody tissue, but they're not inside a vascular bundle together. So these really large structures here are xylem. And some of this other stuff is smaller diameter xylem, but these are good examples of xylem conduits. And then these other kind of roundish structures here, those are phloem. So again, separated in the root, but a little more closely located than in the woody tissue. Okay, let's talk about the structure of the phloem itself. So there are two main types of cells that make up the phloem. There's the sieve tube members, which are these larger cells here, and then there's these companion cells. And then there's these connections between the cells. These are referred to as plasmodesmata, and I think I also talk about this in the next video. So that allows the cytoplasm, which is the liquid part of the cells essentially, to move from one cell to another, and they share that xylem. And then similarly with the companion cell, you can see lots of these areas where those cells are connected through the plasmodesmata. The other thing to notice is these pores on the end of the cells. So this is referred to as a sieve plate. And so there are pores in those plates. And I called these plasmodesmata, but that was, I guess, incorrect. I got ahead of myself. These are just showing connections between these sieve plates from one cell to another. And then you can see there's lots of other structures inside the companion cell and the sieve tube members. You've got endoplasmic reticulum, where a lot of respiratory processes are going on. You can see the mitochondria in here in the cell. They might have chloroplast in them, and then of course a nucleus. And then here's just a couple other images so you can see this in non-cartoon form. So in this upper one, it's a scanning electron micrograph of a sieve element here on top. And then this slightly smaller cell here is the companion cell. And you can see a lot more structures inside the companion cells than in the sieve element, but there's some in the sieve elements as well. And then here's just some more images so you can see the sieve plates. So this is kind of a far away, relatively far away view of a sieve plate. The black circles then are those pores that allow, movement from one cell to another. And then the grayish, the lighter gray areas here, that's just a membrane tissue. And so that would prevent flow through the membrane, or at least rapid flow, and then all of the flow would happen through the pores. So then you can imagine that both the size and number of those pores is going to have an impact on how fast sugars and other compounds can move through the phloem. So if you look at these two panels, panel E and panel F, you can see they're at the same magnification, but you can see on the right we've got fairly large pores and very little membrane separating those pores. The one on the left, there's more membrane space and smaller pores. So those are two different species and slight differences in the structure of those sieve plates. Okay, let's talk about the mechanisms of phloem transport, or at least how we think it works. So this hypothesis was posed in 1930 by Munch, and I'm sure it's pronounced wrong. There's supposed to be umlauts over the U. This was a German scientist. And so the proposed mechanism is that we know sugars are produced in the mesophyll and palisade layer in the leaves of plants through photosynthesis. And so those sugars then are transported to those companion cells that we just looked at. Then within the companion cells, there's kind of machinery that will load those sugars into the phloem. Now here's where the introduction on water potential comes in. So remember that if you solutes in water will reduce potential energy and make the water potential more negative. So as sugars are being added into the sieve tube element, it's lowering the water potential in that cell. As water potential gets lowered in that cell, water is going to move from places of relatively high water potential into that sieve tube element. All right, so we're loading sugars into the sieve tube element as a way to draw water into it. And as water moves into it, that cell kind of expands. Now remember, in the water potential equation, we've got not only solute potential, but pressure potential. And remember, in a plant, that pressure potential is created by the positive pressure of the cell wall back onto the contents of the cell. So as the sieve tube element fills with water, then it's pressurized, much like a water balloon, right? So you fill up a water balloon, and if you don't tie off the nozzle part, water is just going to shoot out of there. Similarly, as you fill up the sieve tube element, it's going to want to push those contents away to places of lower water potential. So that's how the water is moving. We pressurize essentially the sieve tube element at the source, and then cells at the sink. Let's go to this diagram. I should be using this diagram. So again, we've got lots of sugar molecules in the sieve tube element at the source. It lowers water potential. You can see in this diagram that water is moving from the xylem into the phloem. Then if we go down here at the sink, the companion cells at the sink are removing solutes and sugars from the companion cells. So that raises water potential. Water moves from the phloem back to the xylem so that the pressure potential goes down. So the cells at the sink aren't fully expanded with water. They actually don't have that much water in them. And so then that's the high pressure at the source is pushing contents down to the sink where those sugars are being removed and water is moving out of those sieve tube elements. Okay, I jumped too far. So that's where I'm going to stop. Just general mechanisms of the phloem transport. The next video will talk a little more about some of the details of how sugars are getting from the source into the companion cells and some of the implications, ecological implications of that.